I don't.
Thanks for coming. We're live. Welcome to Science on Tap. Thank you for coming tonight. My name is Carol Warden. I work at UW Trout Lake Station. Science on Tap is an example of the Wisconsin idea conceived in 1905, where the borders of the university are the borders of the state. Our partners in Science on Tap are Rocky Reef Brewing Company, the Lakeland chapter of the Wisconsin Alumni Association, UW Kemp Natural Resources Station, UW Trout Lake Station, Monaco Public Library, and WXPR Radio. Science on Tap is also supported through a grant from the Brittingham Fund from the University of Wisconsin-Madison. You have 3 ways to watch. You can join us right here in person, like many of you are. You can watch live on YouTube through our website, scienceontapmonaco.org. Or you can watch events later, and they will be archived through our website as well. Our next event will be Thursday, January 4th, and we'll be hearing from Bart Kotarba and Amanda Shermer from the Northwoods Wildlife Center right here in Monaco, talking about the wildlife of the Northwoods. Tonight, we have an impromptu guest speaker, Dr. Gretchen Gerrish. The speaker we had lined up had a family emergency, so we went in-house to get somebody to fill in the last minute, and Gretchen was up to the task. So, thank you very much. I also wanted to let you know of another event that is going on January 11th at the Monaco Public Library from 6 p.m. to 7 p.m., and that is Science on Tap and the Wisconsin Academy are hosting an event with artist Mary Burns. She's an artist in Manitowish, and she does jacquard woven tapestries. And so she will have an event of the tapestries that she has made representing women and water. So that's January 11th at the Public Library. Tonight, we have Dr. Gretchen Gerrish. Dr. Gretchen Gerrish is an evolutionary ecologist that studies unique life histories of aquatic invertebrates in both marine and freshwater ecosystems. Her work focuses on how reproductive behaviors and habitat use influence the rates and patterns of divergence. I'm sure she will explain what that means. Dr. Gerrish studies bioluminescent ostracods that use light for both defense and courtship displays. Gretchen is a teacher, a mentor, a researcher, administrator, friend, sister, wife, and mother. She is currently the director of our very own Trout Lake Station, and she believes that nature-led inquiry and training are essential for building understanding and new research projects. So, for our tradition, we have a trivia story about Dr. Gretchen Gerrish. Gretchen has spent a lot of time researching these bioluminescent ostracods in the waters off the coast of Belize. For scientists working in these hot, humid settings, it's become sort of a rite of passage for a uniquely tropical experience to occur while putting in long hours in the field. While these experiences aren't necessarily pleasant, maybe far from it actually, it's something that becomes bragging rights for these scientists. So, what happened to Gretchen that she can now claim as having happened to her thanks to her commitment to her work? A monkey stole her gear straight from her hands while she was setting up on shore. B, botflies burrowed into her skin and laid their scaly eggs in her flesh. Or C, a barracuda charged her and her shining flashlight during a night dive, resulting in a toothy scar across her forearm. Botflies! B! Dr. Gretchen Gerrish.
6 at one time. It's a rewarding experience that is better told over a muskie bite. Thank you, Carol. As Carol mentioned, I am really excited to be able to step in today. And when I was invited, I thought, you know what? I talk about the lakes up here, the ice dynamics, winter is coming. And I love sharing things about the lakes and bragging about all the great work that the Trout Lake Station is doing. Got to co-present with Scott Bovey a couple times, but this provided an opportunity for me to transport you all down to the warm waters, the sandy beaches, and a little bit of salt, and to share a system that I've worked on since 2001 when I started my dissertation work, I first got introduced into the luminescent ostracod system. I was invited down there by my mentor, Jim Morin, to work out the life cycle of a group of ostracods that had been newly discovered. And they're about 3 millimeters in size. And so when I started the project, I went down, and like many crustaceans of that size, we thought, oh, it's probably going to be 3 weeks, a month of work to figure out how long these guys live, what their reproduction looks like. They lived for 9 months. And there we were. So a year later, I finally finished up their life cycle. But during that learning, was really drawn in by a ton of the different types of questions that we could ask and study in this really cool system. So my goal today is to introduce the group, talk a little bit about biochemistry, life cycles, and really amazing behaviors to inspire you to think about these unique systems and hopefully someday head down and take a look into the dark waters and explore. And then I'm also going to use it as a stage to discuss how artificial light can influence many of the organisms in our natural systems. For bioluminescent organisms, it has an especially big implication. And so that's kind of the roadmap of what I hope to share today. So what is an ostracod? It is a tiny, I think Amber around is, yeah. So Amber actually has some examples. It's a small organism. Usually they range from 1 millimeter to 3 to 4 millimeters in size, so microscopic. A lot of the crustaceans I work on are microscopic. And ostracods can be found in all the world's waters. Actually, in the puddle in your driveway, they're one of the first groups that would show up. So you can find ostracods in those waters. There's species that live only in the wells of our pitcher plants or bromeliads. And so ostracods really are a broadly distributed group that were some of the earliest crustaceans to evolve. In addition, they're also one of the most studied. In freshwater systems and shallow marine systems, they form a really strong calcium carbonate exoskeleton or shell. And so they were especially prolific and diverse during the Carboniferous. They have a very good fossil record because of their shell. And who cares a lot about fossils during the Carboniferous? People looking for oil. So a ton of fossil ostracod and evolutionary work in this group has been funded by industry, which is kind of an interesting driver of science that then leads to a ton of additional work and discovery. The group that I work on is actually a divergent group from the freshwater groups. It's a strictly marine group called the myotocopids. And most ostracods do not produce light. But one lineage broke off from all the others and evolved the ability to produce light. And the original evolution of this was as part of a defensive mechanism. So when an ostracod gets eaten by a larger organism like a fish or something, it will express all of its light, and it'll turn in, you can see it from across this room in a clear ocean. And some have phrased it a flash bomb defense. It'll light up, and the fish panics. It 75% of the time, it'll spit that ostracod out. It might taste bad, but also there's a hypothesis that that fish, the entire outline of the fish can be seen, and that it could draw in a bigger predator. It's like that Star Wars scene that says there's always a bigger fish, or there's always a bigger... So we're still studying that dynamic. That we call that a burglar alarm hypothesis, that they put off an alarm and the burglar's lit up and can get caught by the big fish. But in the Caribbean, this light has actually evolved as part of reproduction or courtship. And so all throughout the world's ocean, you can find ostracods that light up for defense. But only in the Caribbean have we observed this really cool, really unique, and really awe-inspiring bioluminescent behavior for courtship. And so ostracods, like I said, all throughout the ocean, 3 millimeters in size, the size of a little pinprick, but they can produce light that could be seen from across the room. Luminescence, or light production in biological systems, has evolved many different times in many different organisms. I think it's over 40 different species have evolved the ability to produce light as part of their strategy for about 75% of the evolution of light production is for defense. especially in the ocean, this is a really common reason that light production is evolved. A lot of the chemicals that are involved in the interaction producing light are relatives to the chemicals that you find in the digestive system. So often it's kind of a vomit up of chemicals, or recently described as snot, luminescent snot, sexy luminescent snot that the ostracods produce. But it's a chemical that's related to other enzymes that are in the system. And so the ostracods, like I said, luminescence has evolved many times, and most of them have to do with luciferin and luciferase. So there's a protein called luciferin and an enzyme called luciferase. And when I describe enzyme proteins, what I think of is that enzyme is like a giant Pac-Man, and it stays intact. And the substrate is like a delicious pizza pie or little piece that gets broken up when that Pac-Man hits it. Your Pac-Man stays intact. So if you have a big enzyme and a tiny piece of substrate, you're going to get a teeny tiny burst of light. Versus if you have a big enzyme and a giant piece of substrate, it'll take a long time. You'll have a long, drawn-out, very bright light. It's these ratios that allow the ostracods to evolve short pulsing lights or really long lights. And so Amber was handing out a little prop. I don't know if folks have their little sticks. So the chemicals that ostracods use are comparable to those that you find in other taxa, except so they have this base luciferin molecule and luciferase molecule, but the tail ends of that can be different shapes. And that means in some groups, the light they produce is red. In others, it's yellow. In others, it's green. You've probably seen fireflies around here. What color would you say? Yellow-green, right? That's kind of the observation. A lot of fish do red, and the ostracods that I work on are very blue, although they have slight differences in the wavelength among the different taxa. And so when we think of this reaction, the glow stick that you guys have works on a similar principle. So the light is a chemical reaction where it's an oxidation reduction reaction where the energy that's given off, like many chemical reactions give off heat energy or light energy, this is a cold light energy reaction when those two chemicals mix. And so in glow sticks, maybe you've all thought about this, but it's kind of fun to think about how it works. Just like the ostracods, glow sticks hold two different pockets of chemicals. On the outside is one chemical, and then inside there's a glass tube that when you break it, allows the other chemical to release. And so my demo here is to get you to think about the ratios and the speeds at which these things mix. So if one of you if you get in kind of a pairing, and one of you breaks your stick and just lets it kind of ooze versus the other shakes it up, you'll see that it's the ratio and how fast those things mix to create kind of a dim light versus a bright light. We've got a fast demo here, but go ahead and break your glow stick. Ben over here, he went full out. So one shake and the other just watch it kind of slowly diffuse. Everybody shakes. Oh, I see a really slow red one kind of coming on, gradual. Awesome. So now imagine a tiny 3-millimeter crustacean swimming through the ocean. It's kind of like you swimming through a vat of peanut butter in terms of the size ratio and the friction that you would encounter in the water. These guys can swim the equivalent of an entire football field. Like if you were an ostracod, it'd be like you swimming a football field in less than a second through peanut butter. And then trying to spit, right? Just smooth along your face there and smear. So they have these really crazy behaviors where So the males of the ostracods are the ones that actually put out the light. And what they do is they spin in a spiral, and then they have a stop point. So I feel like they go, whoop, whoop, and they spit this light out. They have an organ inside their body with 2 pockets. And when they spit, they compress that organ. The chemicals come out, and they actually mix in a pocket in the water. And then they hang there. And they mix fast, or they mix slow. And so, again, those are the things that, when you go out to the reef, allow you to see differences between the different taxa. So that's as far as I'm going to go with the biochemistry. I just wanted you to think about it, hopefully have a little tool to help you visualize it. But the next step is taking this into the ecosystem, because it still remains one of the most awe-inspiring sights I've ever seen in nature. When you swim out, so they have a very specific time regime. What I consider kind of my home site, where I've done a lot of work and built a lot of relationships with folks that work in those waters, is Belize, kind of central coast of Belize. And I can predict the ostracod start times there within a minute. They start very sequentially each night. And so let's say we go out in the dark and we swim out, and you're floating over a dark reef right at nautical twilight, which is about 45 minutes after sunset, and it's considered the time when visibly you can't see the horizon. And you start seeing the first species that go, we call it the slow vertical upward species, because it puts out these big pulses of light that slowly go upwards. And one male will start the calls, and then multiple males around will start going, to where in a reef, kind of this front audience size, you'll see 100, 200 going at one time. The next species to start on that reef is one we call the rocket. It starts way down in the coral, and then it shoots up really fast, like a rocket. That's why we call it that. The next species to go is actually one of my favorites. We call it, its new name is Maristella chicoi. One of my students recently described this species for the first time scientifically. And it goes horizontal, and one male will go in a row, putting out teeny tiny short pulses of light, and then other males will come up and intercept it and do their own lines. And so it turns into this radiating kind of fan of light as it goes out, and you'll have one radiating fan kind of underneath another and underneath another, to where it looks like a waterfall from above. Really incredible male behavior in relation to each other. The females are hanging out in the sand, come up and intercept the male for reproduction. And then the next species that goes, also a fun one, really one of the bigger taxa, and they do these quick pulses going down. It's like flashes going off in a theater or something. It's like poof, 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 poof. And so just on a reef, kind of the size of this room, you would see 6 to 8 species, and they'd have this staggered start. And they'd all kind of peak at about 35, 40 minutes after start. An hour later, you don't see any lights. It's a very brief window. And I think this is one of the reasons why a lot of folks have never been able to observe this behavior, is because it's about 6 o'clock to 7 o'clock, maybe 7:30, depending on where you are in the year or where you are in the Caribbean, but it's dinner time. Dinner time for people who are there recreationally playing around. It's also dinner time for other things that you want to avoid in the water. So that window of time is one where not as much exploration is done, and so I feel really fortunate to have focused a lot of work on that period of time. But if you went out at 10 o'clock at night on a moonless night, you wouldn't really see any of this behavior. So it's easy to miss. And some taxa throughout the Caribbean also only go for a few nights after the full moon, and then they wait until the next lunar cycle. So they're very, very synced. One of my first studies was looking at how they synchronize with the moon and the sun, and their timings are very specific, and some of their windows are very short for observation, which makes research always challenging, especially when it's in faraway systems. So the ostracods, just making sure I hit on the points that I wanted. So that's kind of our reef description at night, some of our active projects in this area. So the system has been known by the Guna in Panama for a long time, the indigenous communities in southern Panama. And even as Chinese vessels would come to the Caribbean, they described lanterns, and they would take these little ostracods to help light up their lanterns. And so I was going to show you, I've got a few examples here. You want to dim the lights for me? I'm going to try this. These are actually dried ostracods that we use for outreach. And so you can see kind of the light. Oh, nice. The light that they produce. I don't know if you'll be able to see it all the way in the back, but I'll give some to Amber maybe to carry to the back. But here in the front, you can kind of see can you guys see that at all? They're like really bright blue. Oh yeah, there you go. Kind of bright blue. Can you guys see anything there? Yeah. So this is that luciferin-luciferase reaction that I was talking. Oh, there you go. Now they're kind of all over. So even dried, these chemicals stay stable, so mariners used to use these to kind of light up maps or in little lanterns in their boats when they were early exploration. The first paper that came out on them by Jim Warren was in the 1980s, describing some of the groups from that Panama system. And since then, it's been really, thanks. Here, I can put them all in there. You're going to spread them around, Scott. There you go. Yeah. Thanks. I don't want to drink that anymore, I think. So the ostracods, they've been known for a long time, but their first description and our documentation of their life cycle. So the first folks that saw them thought they were an algae, thought they were a bacteria. But then when you net the system, you don't get any of those really, really tiny organisms. It's these ostracods that show up. And so those descriptions that happened in the 80s, I was working on things as simple as life cycle and when they're active in the system and what's going on. And so we still have a ton of really basic discovery. We've been doing a lot of phylogenetic trees, asking how they evolve. And one of the really interesting recent findings is that throughout the world's oceans, you have maybe 10 species of ostracods that luminesce for defense. In the Caribbean, in the islands that we have explored, just in the last 5 sites, we found 63 new taxa. And so the hypothesis is that reproductive courtship, kind of those unique signals that each species does, is actually allowing for them to speciate and to diverge more rapidly than the ones that don't have those reproductive behaviors. We also just had a big feature in New York Times last week and Science magazine that highlighted this in training behavior. So that signaling line one that I talked about, one of our students put out a paper, and in Panama we had this really wild, thanks, you can put lights back on, had this really wild observation, went out into seagrass beds, and one of the researchers flashed their light, and the ostracod wave of light would flood from the back to the front. It was literally tens of thousands of ostracods syncing off of one another that would create these waves of light that would wash through the seagrass beds, very synchronous, all of the males going at once. How does the female even see them to choose? Each male display was like 8 seconds, and it was just, right? We have a different perception system, but it was very overwhelming to me. I couldn't even pick out a single line. And so that behavior, how does that even arise, and what is the function of that, was the onus of that study. And so trying to figure out how reproductive success happens in that system. And then the other big topics that we've been working on is thinking about artificial light. And so you saw the light that I kind of had on my hand up here at the front and the back, but we had to turn off all these lights because it was hard for us to see that light. And artificial lights in the marine habitat also change the visual system for these organisms who have their eyes are made up of these little individual lenses and amplify the light, so they are very sensitive to light. And so I was working that little Southwater Key that I work on in Belize. Just across from there is a Smithsonian research facility, and I've worked there called Caribou Key, and they had a couple boat motors stolen. So in 2017, they put in these 3 huge lights projected out on the ocean over their boats for safety. And it wiped out all the ostracods. This was one of the first described groups, wiped them all out within 300 meters of the island. Across the bay, which is about a half mile to the populations I'd been working on, you'd see some of the taxa in the surface water doing these crazy patterns. And I was like, what's happening over here? But it was disruption from the reflection on the surface water. And so that was one observation. Another one, we sampled all the way from Honduras up to Mexico along the Mesoamerican Reef. And around Belize City, for about a 9-kilometer stretch in this kind of circle shape, there were no ostracods. We couldn't figure out. There were perfect habitats, good reefs, good seagrass beds. Really stumped us until you look up. And you think, is the moon up? Right? Like you're looking around. And it was just the glow dome of the city that was really apparent there. We don't have conclusive evidence that that was the cause, but when you're in the water and you look around and you think, I can see everything right now, I think that was a really good indicator that light is a concern. And then we followed up with some preliminary stuff, just working with flashlights underwater and seeing the behaviors. We also had a lunar eclipse, and we're looking at how that influenced their start time or their behavior and interacting. So they're super cute on light cues. They'll go in, if you give them the right light dynamics, they'll start displaying. And so we do know that they're very sensitive to very low levels of light and the amplification of light over time. And, I think I'm sure maybe one of the questions would be like, well, why do we care about ostracods anyway? Which I think is wrong, but a fair question, right? Fair question. But I do think we pick flagship species as indicators, right? It's been a long stretch of looking at polar bears for climate change. And I think that luminescent organisms are one of those indicators that something's off in the system. Corals have evolved to spawn along with lunar signals and solar signals. Most of the organisms in the seafloor depend on those for their reproduction and their success. And that holds true for what we've got going on up here. And so I think we talk about light pollution a lot in astronomy. We talk about it a lot in stargazing. But thinking about how it also has implications for our biological communities. And some of the solutions that we've experimented with in the ostracod system, we use red lights at night, or infrared lights at night that doesn't disrupt their behavior. The red lights do very little. They can't actually see red light. So there's some simple solutions of changing the wavelength to orange and red, orienting just on the boats at the Smithsonian. You don't know how many letters I've written. Just shine it on the boat, not like this. Right, there's some simple solutions that I think we want to continue talking about and exploring. And one of the projects that I hope to tackle up at Trout Lake, I work a lot on the migration of zooplankton in the waters around here. They move up in the water at night to feed where there's less fish predation because there's not a lot of light for the fish to see, and then they move down during the daytime to avoid that fish predation. If you've got a big glow dome or dock lights heavily around a small lake, I think you can change some of that. And so I think it's a fair conversation broadly to think about how lights are influencing our natural systems. So I want to leave time for questions. I feel like I talked a really long time.
We'll have the mic going around with Amber, so please wait till she can get to you so everybody in the room and our audience online can hear you. So you can raise your hand and she'll find you.
I was just curious, you mentioned there's different species, they have different light patterns. Mm-hmm. And have you been able to bring them like in the lab or see if, drive that wrong, to see if they would, since they have specific light patterns, would they breed with each other, or if the light patterns are so specific that these individual species will not only breed with their own species, I guess?
That's a great question. So will the ones displaying differently breed together? So where do we define our species boundaries is really a great question that we've had debate amongst our research group. So the genetics definitely show that they're not regularly mating. So we have that evidence. And then in the lab, the ones that are most different, right, we've got some that, like that horizontal one, the slow upward one, they don't recognize across taxa, but we have experimented with some that are more closely similar, and you see preferences of the females, but I think, so the display is complicated too. I should take this opportunity to describe it. So the first few pulses of a display are usually what we call the initiation phase, and it's the male puts out like boom, boom. And when that happens, one male is putting out the light, and other males see that, and depending on how far they are, sometimes they'll swim over and they'll sneak on that male, and they'll cycle next to it, to where you can get like 40 males around one line of lights. And so then the females will respond both to the lights, but also to that ball of males that are cycling up. So between species, we do sometimes see females like, what's going on? It looks like a ball of males over there. So the light versus the kind of cluster thing, it's a little, yeah, I think the researcher Nico said with a discoteca, right? It's like all of a sudden the disco starts and you're like, whoa. So they do have some interspecies response, and we are experimenting with if that's more common if they're closely related or more likely if they're more diverged. So it's a great question. Hard to tease apart. It's also hard to tease apart because females, I talked a lot about the male behaviors, females are very shy. They come out, they molt into their adult instar and become reproductive, and then they get into the mating arena. They mate, and then they brood their offspring for like 40 days. And so females probably only get 1 to 2 bouts of reproduction in their lifespan. And so all these males are competing, and the females come through and only get like 1 shot. So that too, it's hard to get receptive females in the lab to experiment with. Yeah, but we've done it. It's just a lot of still left there. So, yeah.
So if a person were to go to Belize specifically to see this, when would you go, where would you go, and what other things in the water do you need to watch out for?
Yeah. Yes, great question. So we always go right around the full moon, which seems weird, right? But the full moon, so the night after the full moon, the moon rises about an hour after sunset. And then the next night, the moon rises about 2 hours after sunset. And so right after the full moon, you get full darkness at sunset. And the other thing is those females accumulate for a few days. So right after the full moon, you also have some of your best density of male displays. So there's 2 weeks after the full moon where when the sun sets, it's dark. And that's the window. Because otherwise, if you go at other times and the moon is up, you have to wait for the moon to set. So then you're going out at like 1 or 2 in the morning, which gets a little tougher. So I always say a couple nights after the full moon is going to be your best bet. And then you got to go out right at that. It's really right at nautical twilight, the end of nautical twilight, which is 40 minutes after sunset. Wear a full skin, cover your body. The nice thing is that these guys are in shallow habitats, so we do a lot of snorkeling and scuba diving, but they kind of max out at like 30, 40 feet. There's other luminescent stuff down deeper than that that's cool, but the ostracods seem to really not go much deeper than that. And so it is always good to wear a full skin at night in the ocean. The biggest risk is jellyfish, some box jellies that'll give you a big sting. In the Caribbean, the good thing is we don't have the nasty Great Barrier Reef ones that can do big damage, but it still does not feel fun if you get stung by a box jelly at night in the ocean. I've run into lots of other things that people might feel are risky, but I think really there's not a ton of other things that are going to come and get you. The other thing that's weird is if you have your flashlight, fish are attracted to light at night. And so there was one student that came out with me that had a headlamp, like a light attached, and so the fish would like bang in their face. So you always want to make sure your light is not by your face, and if a big fish is coming, you realize that they might just be coming for your light. So did that answer your question?
Time of the year?
All year, yeah. We've seen them at all months of the year in Belize. And pretty much most of our sites, I think we've seen them pretty much year-round. It has a lot more to do with the moon in its phase, so yeah. And snorkeling is great for them because of this view. You get a big, wide view. Diving's also good, but not required. So that's the nice thing about it too. Yeah. Yep.
So Gretchen, it probably takes some energy for the male to produce these enzymes and do these displays. I mean, how long can they do this? Do they kind of have like one display a night or multiple or?
They can go. It's pretty impressive. So they can produce tons of displays in a night. We really haven't exhausted them in their displays. The light that they put out if they get bit by a fish, that big pulse of light, they basically like vomit it all out, and that you only get like 1 or 2 of those before the male, before they can't rejuvenate enough for that night. But display-wise, not. They have stores. Probably the swimming actually is more energy than the chemicals that they do for the light production. That's our understanding. She's going to grab the mic for you.
I was just going to ask you if you've watched them from the surface and then maybe 15 feet down, is there much of a, like, is there one way that you enjoy watching them or are able to study them better?
Yeah, so for the species display descriptions, we really have to get down and kind of to where we're looking at them as like out in front of us or in it. If you're looking from the top and something's coming up at you, it's hard to judge distance and that kind of stuff. So we do a lot of our description information from diving in the water kind of set out. And we do a lot of underwater video. The cameras that we use for this are really interesting. They're very low light. Even though that light seems bright to our eye, or if you look at your glow stick, the cameras back in the day, so when this first started, the camera was called the Pig Dog, and it was 75 pounds and like this big, and it had a VHS recorder and a Department of Defense night vision adapter, and so you had to position it, and it had no buoyancy control, so it was like wrestling a bubble underwater, trying to get it level out. And then you'd see this one like boom, like green 5-pixel dot. But I really respect the development of that technology that we built from. So that was the original. And then since then, we've evolved that technology. And your camera, our regular cameras, even some of the better cell phones right now, you kind of film in this ISO range of maybe hundreds to 1,000. The camera that we finally developed that can capture it, the Sony A7S was the one we were using back then, but they've even grown since then. It films up to 600,000 ISO. I don't know if there's any picture takers, but like everyone can see that it's like 600 times more than what we used to be able to capture. And so yeah, it's kind of amazing that how the technology has built with the science. Yeah, so the question is, what depth do we film at? And really, the ostracods determine that. So some of those taxa are right up on the crest of the reef, so they might be up in, 10 feet from the surface. Other ones will be right along the sandy bottom, so then we might be closer to 30 or 40 feet down trying to track them as they swim along, that kind of thing. So. Yeah, the longest display that we have documented went about 20 feet. So the male would start down here, and then it was these slow pulses with like 2 feet in between as it swam upward. So that one was fun to film. You kick up, and come down. So, yeah.
I've got an online question here. Is there a frequency color that is dominant across animal bioluminescence? Most reports are near blue.
It depends on the system. In the marine system, there's definitely a lot of blues. And so when we think of some of the dinoflagellates, which are like a little unicellular algae, and if you've ever been to like the Puerto Rican bio bays, or that's like a little algae that lights up, and it's in a similar wavelength to the ostracods. The bacteria that light up fishes can also be blue, and ostracods. But I think in terrestrial systems, like fireflies, we see a lot more yellows and greens. That's true of our local jack-o'-lantern fungus also, which produces bioluminescence. That's more of a green shade. And then in the deep ocean, there's a lot of red. So kind of depends, I think, a little bit on the habitat and the group as much as anything.
Where do the females lay their eggs, and what creatures would eat their eggs that they,
Yeah. So the question about where the females lay their eggs. So the life cycle, the female actually carries her eggs. So she lays them, but she keeps them in a shell on her back. And so she swims around with them in her little pouch so she has them in her body for about 15 days, and then in that pouch for another 15 days, 19 days. And while they're in that pouch, they grow into little juveniles that can crawl around. And so she broods them in that pouch, and she has this crazy, it looks like a comb, and she sits there and aerates them and combs them and keeps stuff off of them. So there's not a ton of predation from big things, but there's this really cool brood parasite. And the brood parasite is wild. I've watched it in action, and what happens is if a female has it, usually it comes in and sits in her carapace or her shell, and it sits right up here. And it's an isopod, so it's another crustacean, but it looks like a big blob. It doesn't look like a crustacean at all. It sits right here as a little blob. And then as the female lays her eggs, it starts eating them, like almost simultaneous. And it eats them, and it converts them into its own egg masses. And so it'll produce an egg mass exactly the same size as the ostracod egg, but it has like 20 to 30 little parasites in it. And so it'll replace her eggs simultaneous with her laying of them, and then she'll spend the next 2 weeks fanning and caring for the parasite eggs. They hatch out, when she's ready, they hatch out as these little tiny crustaceans and go and take residence in a new female. And we don't know a ton about that. There's been one description. We don't know a lot about their parasites, but that's the main source of mortality for the little eggs, or for that early stage. Yeah, it's a great question.
So I'm wondering, as far as going out and studying them, how much do you have to consider the tides and like the currents, and does that have a big effect on your studies, or?
Yeah, tidal exchange can. So these guys don't like flow. And so the habitats that we're working in, even though we can get into some wavy action zones, they don't like when the tide exchange is really strong. And so you don't actually find a lot of ostracods in high tidal exchange area. I'm going to rephrase that. You don't find a lot of the luminescent displaying ostracods in those shifting waters, because they start coming up and it's like, whoa. And so once in a while, if you have a big storm come through and you get a big like wave action or exchange event, they shut down for a couple nights, or they start trying and then they're like, whoa, I'm not doing this. So we have seen that, but I do think that the habitats you find them in are a little bit sheltered. Yeah, and the Caribbean, the tide exchange is also maybe half foot. It's nothing like we have up north, so it's a very minimal tide exchange in the reef systems of the Mesoamerican reef. So nothing like up north where it's meters at a time. Yeah.
You mentioned these are species that hang out in reefs and shallow water. Are you seeing, or is there research being done on changes in population numbers or distribution as sea surface temperatures change, or ocean temperatures in general?
I would say not directly. We have observations, but in terms of that being a central focus, I'd say no. The bigger thing would be some of them are specific to certain coral types, and so that association with certain coral types is a big factor. So when we see corals disappearing or recovering in systems, we've definitely seen some taxa shift. I think it's a great question to pursue, but a big one and cool. The other evidence that we have of some of these big environmental hurdles for them, Puerto Rico had a major hurricane event, as many of us know, and there was a diverse suite of ostracods in that area, and the sedimentation and the sediment lifting and coral die-off that happened in Puerto Rico, the taxa there have a really different distribution than the other parts of those kind of chain islands, and so I think hurricanes are another interesting thing to investigate in terms of what that disrupts, and because it changes visibility for years when you get that much sediment stirred up and corals dying and seagrass loss. So I think there's lots of room there. Yeah, for sure.
I'm just wondering, is there a website we can go to look at some of this bioluminescence that you've been filming or other people have filmed?
Yeah, I would love that. Like I said, our coolest footage just came out in the New York Times last Wednesday. So there's a new article, but there's also a former one called Sparkling Seas that has a lot of really good footage and a big poster that shows the ostracods and dynamics. I wanted to respect our PowerPoint idea for Science on Tap, but yeah, I'm also happy to share if you email some images, but maybe we can post a few links on the Science on Tap website or send it out to the mailing list, because the visuals really are really incredible for this group. So I'd be happy to share some of that. Or better yet, invite y'all down to Belize to see it firsthand. It can be really hard and a little bit scary to motivate to go into the water at night like that, but it's very worth it. So.
I have a question. I know that this is all about Belize and bioluminescent ostracods, but the whole point about other light in our environment playing a role in their behaviors, and the little tiny tidbit you gave at the end of your talk about how that affects our zooplankton in these lakes, I wonder if you wouldn't mind diverging just a bit and giving a little bit more detail on that.
Yeah. Well, I think it's really common. This is just so I am diving in to the freshwater light exploration, but I think that it's really easy sometimes to see the impacts of light on the large characters in our habitat, right? Folks know it for sea turtle hatching and the orientation of those offspring and spawners. Frogs calling around here, you probably recognize bats swarming the lights. Why do they do that? It's insects, right? It's because they're after the bugs. And so things like zooplankton and these migratory behaviors are a daily cycle. And this is the group that feeds all those big things. Just like on land, the insects are really the feeders of all those big things. And so zooplankton have evolved this migratory behavior. It actually represents kind of the largest biomass migration on Earth when you think about all these tiny things moving up and down in freshwater and the ocean every day, sometimes hundreds of meters. And so we've done studies on Crystal Lake nearby, a preliminary study with Julie Bruskirk in 2019, where you put a big light on the boat and you sample how many zooplankton are in that surface water, and then you sample nearby where that light wasn't. And you see that there's many more zooplankton when you don't have the light, but you also see that the types of species that are there changes, right? And so that's a pilot study, kind of preliminary. But there's a lot of logic in thinking about how this disrupts. And I think there's also been a ton of change in the application of these solar lights, right? It used to be that you turned lights on and off. And we would put our dock lights on, you turn it on when you're out there using or recreating. And now solar lights are great because you don't have the same energy use and more environmental in that way, but they also don't go off at night. They stay on often throughout the entire evening. And so not as bright. I think there's a lot of exploration. The other thing is if solutions can be just a change in the wavelength that's still appealing to us or useful for safety, and minimizing that, I think, is a big deal. And there's places throughout Wisconsin that are claiming dark skies. And I was here in the 90s. Crystal Lake's one that I had frequented back then, too. And it's amazing, because back in the day, there was an event where we had all the campgrounds went out, the fires went out around us, and it's a perfectly round lake, right? And so you're in the middle of the night, it's dark, the fires all go out, and it's all smoky. And you look up and you say, I don't know where I am. And you got to row in some direction, totally lost. That's back in the 90s. And today, if I went out on Crystal Lake in the middle of the night, which I have recently, I'd be able to say, oh, there's Minocqua, let's go that way, right? Oh, there's Boulder Junction, I'm going to go that way. It's changed. And so I think that there is room to just make sure that this isn't an additional pressure that we're putting onto systems when they face so much anyway. Thanks. Yeah.
I've got an online question. How are these similar or different from fireflies in terms of bioluminescence and their behavior?
Yeah, and sometimes people call them sea fireflies. That's definitely a parallel that folks make. The color's different, so the blue versus green. So the light production in the two groups evolved completely independently. They both evolved a luciferin luciferase system, but it was different. So the side chains are different. That's why the colors are different. I think some of the other big differences is most fireflies have a male call and a female response. And so most all of the fireflies have a back and forth dialogue and then they merge. In the ostrich system, it's really just the males that call. We haven't seen good evidence that the females have a light response. There could be something chemical in there, right? But we don't have that yet. We don't have any documentation, and so that call response thing isn't evident. And then the other thing, two other things, the sneaky males, right? Ooh, there's so many. Like one light string, again, 30 males, like congregate on that. And there is sneaking in the firefly system, but not at that scale. And then the other one is just the number of species that coexist. So the kind of highest density for fireflies, kind of at the margin of a grassland forest along those edges, is about 4. And on a single coral reef, just the size of this room, we could have 6 to 8 regularly. So that kind of overlap in the habitat is probably another thing that I think of being a little bit different than what we see in the fireflies. You could have 50 ostracods displaying over just this bench. It's like incredible how dense they can be. Yeah.
You might have answered this earlier, but that swarm of males that goes around the light, is it kind of one of those reproductive strategies where they can't produce the light, and so they're hoping that an intercepting female will hit them on the way to the light? Or what's kind of the strategy behind that?
Yeah, that's a great question, Cheston. And I love it because I don't think I did the description of that sneaking behavior justice, because this mating system, it's what we call a lek and spree. If you've thought about mating systems, what that means is that all of those males are capable of luminescing. So all of them are reproductively active and capable of doing a display. And so as that male does those initiation pulses, the males, if they're really close, they'll sneak. So if they're within a certain distance, they'll sneak and they'll join in the ball with the hopes of intercepting the female. If there's a little farther away, they'll start doing their own display. And they can synchronize or entrain. But I think of this as like a sneaking tactic and this as a distraction tactic. But the ostracod reproductive system is one very different from others in that males can go from being a displayer to a sneaker to an entrainer. They alternate between those 3 behaviors readily. It's not like they're a fixed sneaker or a fixed displayer, which I think was kind of the gist of your question. In a lot of systems, you have like sneakers, and that's what they do, right? They sneak in, they steal the nest, they do the thing. But in this system, it's like alternate mating tactics readily, like one right after the other during the period. So, yeah.
Just a quick question. I was wondering how many species are up here with ostracods? Or if it's even...
Yeah. The question was, how many species of ostracods are up here? To reiterate, there's no luminescent ones, right? And there's actually no luminescent organisms in freshwater. That's a evolutionary challenge we still explore. But in terms of freshwater ostracods, Carol Warden also counts zooplankton. Maybe that's what I'm looking at. Living taxa. I don't know the answer to that off the top of my head. My guess is it's like, I don't even want to put a guess out there. More than 20, less than 1,000. I'm so ashamed right now.
I think that's a great answer. That's about what I would have said.
It's a big old I don't know. But, yeah.
So maybe to end it, a quick bit of opportunity for self-promotion. Gretchen has an IMDb profile and has done some documentary work, and I wondered maybe if you could tell folks a little bit about that documentary work you've been involved in and where they can watch some really cool stuff to see more about it.
Yeah, thanks, Noah. Yeah, that would be one of the links I would add for sure, because it was really science and art and communication. I really am into like how we put those things together, which is why this program is super fun for for me to be involved in. So in 2016, we had applied for a big grant to go and explore a bunch of new ostrich populations, and I reached out to a filmmaker, Martin Dorn, to help build our research cameras. He did that like geothermal thing with lions attacking wildebeests in the first Planet Earth. His whole idea is how can we revisualize nature? And my advisor had a connection with him back in the 90s, helping build this camera and think about a system. So we reached out and he said, oh, I just put out a documentary for luminescence that I really want to get moving, but I'd love to help you build cameras. So we got him on board to build our cameras, and then his film got funded. And it was like, oh, those cameras are going to be great, right? Because now we have two purposes for them. So that turned into a really fun partnership where Martin and his crew came out and filmed the ostracods for 2 weeks when we were in Belize, and then he came back and after they had kind of finished filming the marine components, came back to join us in Roatan, which also has really good ostracod displays, Honduras, and film that. And so it was a really cool collaboration, lots of back and forth and dialogue. We talked about ostracods. There's some luminescent fish that are in there and these cool sea stars that luminesce that we got to share with the group. And so they put it all together into a documentary called Life That Glows, or Light on Earth, and it was narrated by David Attenborough. You might see a familiar face and some of our partners on the project. And so that is one of the questions. I do some SciComm like talking, and I think one of the questions I love leaving people with is, if you had the opportunity, which would you choose? Describing 63 new species of luminescent ostracods from all throughout the Caribbean, or sharing a 5-minute story of those with millions of people throughout the globe. And I think partnership with people who really know how to communicate well gives the opportunity to kind of do both of those. So yeah, Life That Glows. You can usually find it streaming. I think right now it's on HBO still, and so it's a fun one to watch. And I'll put a link to that on the website for folks. But thanks, Noah.
Yeah.
It's fun. We also had clips in the recent Disney series, and what was the name of that? Anyway, I'll put that link up there too. But it's a pretty cool system that people like to see. So it's gotten a little traction that way. But thanks for the.
Well, thank you. Join me in thanking Dr. Gretchen Gerrish. I'm going to pop up here with you. Thanks to all of our partners, UW Trail Lake Station, Rocky Reef Brewing, Monaco Public Library, WXPR Radio, Kemp Natural Resources Station, and the Lakeland chapter of the UW Alumni Association, and the Brittingham Foundation. We hope to see you all back here in January for our talk on Northwoods wildlife. And don't forget about our January 11th event at the library for Mary Burns' Jacquard Woven Women in Water. If you want to sign up for any email reminders, you can do so on our website, scienceontapmanaqua.org, and click on Contact Us. See you next month.